Hi, my name is Tiagi, and in this video, I'll be going over AP Bio Unit 2, Cell Structure and Function. So to start, according to cell theory and biology, all living things are made of cells, and within these cells, there are two types of cells. There are prokaryotic cells, so prokaryotic, and there's eukaryotic cells. And the main difference between these two types of cells are that prokaryotic cells do not have an internal membrane, and thus they have no membrane-bound organelles. Eukaryotic cells, on the other hand, they do have an internal membrane, and therefore they have membrane-bound organelles. So the two types, so prokaryotic, there's three domains of life. Prokaryotic encompasses archaea and bacteria. Eukaryotic encompasses eukarya, which include plants, animals, protists, and fungi. And so because these prokaryotic cells do not have internal membrane-bound organelles, they don't have a nucleus, right? Because a nucleus has a membrane. They don't have nucleus, right? Because of that, they have DNA that's unbounded in this sort of nucleoid region. So everything is kind of just unbounded and floating around in prokaryotic cells. Eukaryotic cells, on the other hand, have nucleus, and they're typically much larger than prokaryotic cells. And I guess you can kind of think why, right? Because we have so many membrane-bound organelles, which make the cell larger. Okay? So cells, all cells, prokaryotic and eukaryotic, they both share four common things. They all have a plasma membrane, or a cytoplasmic membrane. And so this is basically just a lipid boundary. So lipid boundary around the cell, like so, that separates it from the environment. You also have a cytosol, or cytoplasmic fluid. And basically, this is kind of a semi-fluid, gel-like material, and it helps to hold all the materials inside the cell together. And it's also site of many metabolic reactions. Next, we have chromosomes. So all cells have chromosomes, which are sections of genes or DNA. And so these sections of genes help to create proteins through the use of ribosomes. So all cells also have ribosomes. So ribosomes help to create proteins via mRNA, which is found through the DNA. And so because all cells have these things, especially ribosomes, right, we have the theory that all cells have a common ancestor because they all have ribosomes and they all have these certain molecules and materials. Next, what kind of membrane-bound organelles are there? So there's many different types of membrane-bound organelles, but the ones you need to know for AP Bio start with the ER, or the endoplasmatic reticulum. There's two types. There's rough ER, which kind of looks like this, and you have some dots on these little folds. Rough ER, what it does is the site of protein synthesis. So these little dots are actually called ribosomes, right? And as we know before, they help to create proteins. So the rough ER as a whole helps to create proteins. We also have the smooth ER, which is similar, but it doesn't have these dots, so it's not rough, right? And smooth ER helps with creating lipids, and it also helps with cell detox, so to kind of detoxify certain substances. Next, we have the Golgi apparatus. The Golgi apparatus has this like bunch of folds, right? These little sacs, and what it does is it modifies proteins for the purpose of transporting them and it packages them. So it packages them, then be transported. So if we imagine a protein created by a cell that's meant to be going to another cell, what would happen is the rough ER would be created, it would go to the Golgi apparatus where it would be packaged, and then it would be transported out of the cell through the plasma membrane and to where it needs to go. Next, we have the mitochondria. So the mitochondria has a double membrane structure, so it has an outer membrane and an inner membrane with lots of folds. It looks like so. The purpose is to take from chemical energy into ATP, which is usable energy for the cell. It does so through cellular respiration. Next, we have the lysosomes, which are kind of like the garbage truck of the cell. And their job is basically to digest waste materials and just digest in general. And they're also involved in apoptosis, which is programmed cell death. And the reason why we have programmed cell death is because eventually cell components just kind of wear down, and so we need to have this kind of mechanism for killing the cell when it's time. And it looks like a little sphere with these dots on the outside. Then we have the vacuole, which stores water, nutrients, and sometimes waste. In animal cells, the vacuole isn't centralized, and so if we imagine an animal cell, the vacuole is spread out. So we have these little dots, those are vacuoles, and it's all kind of spread out. But in plant cells, we have one large central vacuole. So if we imagine a plant cell, the vacuole would be like one large central kind of thing. And because it's like one large central thing, it also helps in keeping plant structure, because it helps to keep the plant rigid through this one large central object. In terms of the differences between animal and plant cells, they're pretty similar overall, but one of the major differences between them that you need to know for APBio is the plant cells have chloroplasts. Chloroplasts are, so while mitochondria creates energy for animals, chloroplasts in plant cells help to create energy for plants. And so chloroplast consists of, again, an outer membrane, and then inside we have these inner membranes, and we have these little sacs that are called thylakoids, and this is where photosynthesis actually happens. Some key things to note about the chloroplasts. So the stacks are called grana, stacks of thylakoid, and the liquid in between is called stroma. Plant cells also have something called a cell wall, which is outside of the plasma membrane, and it just acts as something that helps to keep the structure and rigidity of a plant. So if we look at a plant cell, it's going to be looking more like rectangular, more structured, versus an animal cell, which looks to be more like round and less structured because it doesn't have a cell wall. Next, let's talk about cell size. Let me just erase. So cell size, when we're looking at cell size, we're looking at the surface area, which is given to us by the size of the membrane, and volume, which is given to us by the size of the cytoplasm or the fluids in there. And so we know the surface area goes by n squared, right? Versus volume, which goes by n cubed. And so from this, we can see that volume increases as a faster rate than surface area. Because of this, because volume is increasing at a faster rate, if we look at the surface area to volume ratio, or surface area divided by volume, we can see that small kind of animals or cells have a higher SA to volume ratio because, again, it goes by n squared to n cubed. And so if we have smaller n, so a smaller cell, we're going to have a smaller SA to volume ratio versus large, which have a lower SA to volume ratio. And this SA to volume ratio is crucial because the idea is higher SA to volume ratio means you have more surface area for the membrane, and because of that, you have greater efficiency in exchanging and intaking materials. Okay, and because you have this greater efficiency, you have more cellular efficiency in general. Versus lower SA to volume ratio or large cell, you have less efficiency. And because of less efficiency, most cells aren't really big. We can't have big cells because if we had a big cell, we'd have a huge volume ratio, surface area. And because of that, if we had a molecule that we needed immediately, right, on the outside, it would take a long, long time to get inside, and it really wouldn't be efficient because, again, we have such a large surface area and such a small surface area compared to the volume, which is why we have kind of small cells versus big cells. One more thing to note is that because we have a high surface area to volume ratio, we're also more susceptible to heat loss. So it's increase in heat loss, which is why that smaller animals have a higher metabolism. So like bunnies and stuff, they have higher metabolic rates because they need to generate more heat because they're losing it faster because their surface area to volume ratio is higher. Next, let's go through the plasma membrane and the stuff that makes that up. So the plasma membrane is what separates the cell from the outside environment, and it's made of a phospholipid bilayer. And so we have these phospholipids, and it's a bilayer, so there's two layers. And so some important things to know: these heads, these phosphate heads, are hydrophilic, which means they are polar and they like to be around water, which is why they're facing it, versus these tails, which are hydrophobic and nonpolar, which is why they're not facing the water. And the idea with this plasma membrane is that within this plasma membrane, we can have proteins embedded in two types. So we have peripheral proteins. Peripheral proteins are proteins that attach either on the inner part of the cell or the outer part of the cell. And we have integral or transmembrane proteins, which attach through the cell or through the plasma membrane. So something like this is a transmembrane protein. And so basically these proteins, they're charged, their R groups are either charged or uncharged depending on whether they're the peripheral. If they're on the peripheral, right, they're going to be charged, right, because they want to be attached to the polar section, so they'll be charged. But if they're integral, they're going to have sections that are uncharged, right, to be with the hydrophobic sections. But they're also going to have sections that are charged to be with the hydrophilic sections. So depending on that, they'll be charged or uncharged. Additionally, this model of the membrane is called the fluid mosaic model. And basically the idea is that components are fluid in the membrane, so these proteins are fluid, and they can be moved around and adjusted in the membrane. Next, let's go through permeability of the membrane. So the membrane is permeable, semi-permeable, which means that it's able to selectively intake certain materials and then outtake certain materials. And the reason why is because of the structure. The structure allows it to be selective with what materials it takes in, what materials it takes out. And the idea is, here, the main thing you need to know is that small non-polar molecules, such as N-nitrogen, oxygen, or carbon dioxide, are freely able to enter and exit. Small polar are able to sometimes enter and exit, but they're able to enter and exit slower. So water, ammonia gas, H3. And then finally, we have large polar cells, which are not able to enter the plasma membrane. And so for large polar molecules, if we need to take them in, what we have to use a transport protein. So it's not able to just slide through the plasma membrane, right? Because it's permeable, right? Semi-permeable. It needs to have a transport protein to actually help it go through large polar, and sometimes small polar molecules need this transport protein. So what are the actual methods for membrane transport? How do things actually get inside and outside? So there are two types of membrane transport. There's passive membrane transport, which relies on this concept called diffusion, which is the movement of molecules from a high concentration gradient to a low concentration gradient. And this is all natural, so it doesn't require any energy. And it happens naturally because of this concentration gradient. So the gradient causes diffusion to occur. So there's two types of passive transport in terms of the membrane. We have one that requires no proteins, and this is called simple transport. And so basically, we have the membrane, plasma membrane, right? This simple transport would just cause the molecule, so it'd be small and non-polar. It would just go through in between the fluid mosaic membrane, go through, and it'll just go inside the cell. Versus the second type, which is called channel protein, or sorry, facilitated diffusion, which uses channel proteins. So with facilitated diffusion, we have these channel proteins that go through the membrane, and so then we have these molecules which are able to go through these proteins and then go through the cell. And so examples of facilitated diffusion, we have some special types called aquaporins. Aquaporins are special types of facilitated diffusion channel protein that allows water to go through. And we have ion channels, which are a special type that allows ions to go through. But again, these proteins, the channel proteins, for facilitated diffusion, do not require energy because they're going from high to low concentration. They're passive transport. Versus active transport, which goes from low concentration to high concentration, and you need energy to do active transportation, right? And so a diagram, or sorry, the main type you need to know for active transport is the sodium-potassium pump, or sodium-potassium ATPase. And so basically how the sodium-potassium pump works, which is a type of active transport, you have this kind of protein embedded inside the membrane. And it's currently, if we imagine this protein, right? This is the outer membrane, this is the inner membrane. This is the kind of closed state. And so what happens is we have these sodiums, so we have 3 sodiums that bind into the protein. And what happens is when we add energy, when we add ATP, this is going to open, so it'll open this way, causing the sodiums to leave. And then we can take in the potassiums, and then when the potassiums are inside, the energy, it takes energy for the potassiums to bind to the protein, which then causes it to close, and the potassiums go inside the cell like that. And so the idea is we don't have that much sodium inside. We have a low concentration of sodium, a high concentration of potassium inside, but we're still able to get more potassium because of this pump. So the sodium, this pump, which uses energy, ATP, is able to get more potassium, even though our concentration is from low to high. And that's an example of active transport. Next, let's talk about tonicity and osmoregulation. So osmosis is the diffusion of water from high concentrations of water to low concentrations. So water, through osmosis, because it's high concentration of water to low concentration, means we're going from a low solute concentration to a high solute concentration, right? Because you can imagine if we have a solution with lead with solute versus high solute, there's going to be more water here, less water here, and therefore, by diffusion, it'll go like that. So low solute to high solute concentration, and tonicity describes how water moves in a cell. We have 3 types. We have isotonic cells. This is where the solution is balanced, and so you have the same concentration solution inside and outside, so there's no net water movement. We have hypertonic, where we have less solute on the outside, and because we go from low concentration to high concentration, because there's less solute on the outside, it's gonna the water is gonna leave and it's gonna shrivel up, and so the shell shrivels up when it's hypertonic. And finally, we have hypotonic, which has less solute, sorry, more solute outside than inside. And because of that, the water is going to go inside the cell, which causes it to lyse, and eventually it'll explode or blow up because, again, it's hypotonic, so water is going inside the cell. And osmoregulation is the process by which cells will regulate the amount of water they have, and they do so with the vacuoles. So they have these vacuoles, right, which store water, and so they have these things called contractile vacuoles, which basically release water, which should then go out of the cell or inside the cell, and they can also take in water, which allows it to regulate the amount of water, and then therefore regulate isotonic, hypertonic, and hypotonic levels. Finally, let's go over the purpose of membranes in organelles, membrane-bound organelles, and finally, the endosymbiont theory. So the purpose of membranes and organelles are to compartmentalize the functions of a cell, which then allow for increases in efficiency. So compartmentalize, which allows for increase in efficiency. The examples are mitochondria and chloroplasts. So because they're compartmentalized and they have their own membranes, right? They have folded membranes, which allow for increased surface area to volume, which therefore allows them to better intake and increase their efficiency because they have a higher surface area to volume ratio. And finally, we have the endosymbiont theory. The theory states that because mitochondria and chloroplasts share a lot of characteristics, so the characteristics they share, they both have circular DNA, they both have ribosomes, double membranes, right? So we have the outer membrane, inner membrane, and we have the little thylakoids in there. And because they're self-replicating, right, because they share these characteristics, we believe them to share a common ancestor together, so they both come from the same ancestor. So if we assume this first, the theory goes, the endosymbiont theory goes, that these mitochondria and chloroplasts were once individual cells that kind of lived on their own. But then what happened is that the first eukaryote, so we have a eukaryote, the first eukaryote, it took in, or it engulfed one of these mitochondria, right? And it turns out that the mitochondria and the eukaryote, they both had a mutual relationship, which means they both helped and benefit each other, which is why it's called symbiotic, right? They both helped each other, and because of that, they were able to then reproduce, and this is how cells ended up having mitochondria within them. So eukaryotic cells ended up having it, because it turns out it was a beneficial relationship to have the mitochondria inside for both parties. And then eventually what happened is that chloroplasts also got engulfed, which led for plants to have chloroplasts inside their cells. And that's the endosymbiont theory. So that was AP Bio Unit 2, Cell Structure and Function, and that should be everything you need to know for this unit. Thank you all for watching, and I'll see you guys all tomorrow. Bye.